My name is Benjamin Aribisala, and today my focus is just to run us through some commonly used MRI analysis tools, some of the software packages that are used in processing magnetic resonance imaging. And so the target is to present 9 this afternoon, but I can say that the tools are by far more than this. And these are just the most common ones. I would say some of them I've used before. And then, so I just thought that it would be good for us to be aware of some of these tools. So I will be presenting on FSL, ImageJ, AMID, ITK Snap, and MITK, and O-Series, 3D Slicer, Free Software, and SPM. Before I go to the next slide, I just want to say that, in my own opinion, FSL, SPM, and Free Software are the biggest and the most commonly used of these 9. And they are very, very powerful tools. And I will argue that if you want to do image analysis or image processing and focusing on MRI images, that you should be able, you must make efforts to be able to use at least one of these 3, that is FSL, SPM, and Free Software. Others are very good as well, but these are the topmost 3, in my own opinion. Now, I start with FSL. FSL is a very comprehensive library and of image analysis tools, and developed by the team in Oxford University. So FSL stands out because it allows you to do a lot of things. But I want to say that you can process MRI images, functional magnetic resonance, resting state images, diffusion, perfusion, all kind of images. And these images can come, typically the FSL allows you to import images in analyzed format, but they can even come in DICOM. Even if you have them in Nifty or Nifty.gz, FSL is happy to do that. So those are the kind of images that you can use FSL to process and the format. Now, what can you use FSL for? I want to say that the focus this afternoon is not to actually train in particular on any of these 9, but just to expose them to us, because to train on FSL only will take you about 4 weeks or thereabout, although depending on the use. So I believe strongly that later in future, that we will have the opportunity to pick 1 or 2 of these tools and conduct training on them. So just to say that. So today, I just want us to know about the existing tools or the common ones, and that's all. So what can you use FSL to do? You can use it for image segmentation, for image registration, image resampling, image arithmetic, statistical analysis, and you can develop pipeline, or you can batch whatever you do. In the area of image segmentation, I quickly mentioned that because most of the tools that I present today we allow you to do image segmentation and registration. And image segmentation is nothing but separating your MRI images, focusing on brain images now, to different chunks depending on your target. So for example, I can see image segmentation actually from about 4 different perspectives. One is when you are separating your brain images to the 3 tissue classes. So although FSL will allow you to even segment into more than 3, but commonly you can separate into 3 tissue classes, white matter, gray, and then the CSF. So that's one type of image segmentation, and FSL will let you do that. The other type is when you do brain extraction, which will cause call scraping, and and there is a function called BET in FSL that lets you do that. So that's another form of segmentation, because it's segmentation because you are removing the bit you don't want and retaining what you want to look at. So brain extraction in common, so generally called. So that's another form. The third form is if you want to do subcortical segmentation, that is, you are interested in extracting the subcortical structures like hippocampus, like caudate nucleus, and the likes, hypothalamus and all others. So FSL lets you do that as well. And that's very, very interesting. The fourth is if you want to do, maybe you want to parcellate your images into different chunks. Sometimes you are interested in measuring the cortical thickness, and so these are the 4 different types of segmentation, and FSL can let you do most of this. And FSL lets you do image registration. And what is image registration? It's all about transforming your image from one, space we call to another space. So sometimes that requires resampling. But when you are talking about transformation, maybe you want to rotate, you want to apply share, you want to, magnify your image. So all the processes combined to answer registration, that's image transformation. FSL lets you do that. We typically are required to do that during image processing. And FSL lets you do resampling. You want to downsample or you want to upsample. That is reducing the thickness of your MRI images or, reducing them. So sometimes you acquire, at, low resolution, but for the purpose of processing, you may want to resample. So FSL lets you do that, and you can do so many image arithmetic, like adding 2 images together. FSL will let you do that. You can multiply your image by constant, and then FSL is really home and dry with image arithmetic. And the last in this particular area is on voxel-based morphometry or volume-based morphometry. So sometimes you are interested in looking at voxel and you want to see a comparison between 2 groups of images, and FSL lets you do that. And I'm still going to talk about SPM, which was developed to do that, but FSL lets you do that. And finally, here on FSL is that FSL can run, is typically developed to run on Linux, but it can work on Windows as well. So you can do the installation on Windows now. And so if you have a Linux box or you have a Mac or Windows, you are really good to use FSL. And it lets you, all the processes you want to do, you can arrange them as a pipeline and just batch them, which makes life so easy. And that is FSL for you. This is the user interface. And I put this just to let us know that FSL can actually let you do visualization as well. So you can see the 3 different orientation, the leftmost sagittal, the middle corona, and the rightmost axial plane. So FSL will let you visualize through a function called eyes, so FSL can let you do all this. So that's FSL, 1 down. So we go to the second one, which is image J. So image J is another image processing and analysis tool. It runs, and it was developed in Java. And so it has a very nice user interface, and it can let you do so many things, even by looking at this particular, landing page, you can start to imagine what ImageJ can do. So ImageJ lets you do filtering, smoothing, thresholding, edge detection, and resampling. So all this can be done. I've used the FSL very well, and ImageJ, where I found it to be very, very useful is when you need to do region of interest analysis. So you want to draw ROI, that is region of interest, on a particular structure. Let's say, for example, you are looking at lesion and you want to analyze lesion. So you can use ImageJ. It's very flexible. The use is very, very interactive. And you can use ImageJ for image arithmetic as well. You have 2 images loaded and you want to add them together, pixel-wise, ImageJ lets you do that. So the third one is AMID. AMID is another image analysis tool that is commonly used for processing and analyzing MRI images. So you can use AMID for viewing, for visualization, for image analysis, for image registration. So similar to what you can use FSL to do, and it runs on Windows, and so it's very portable. And I need to just pause a little bit to say that all the 9 tools that I'm introducing to us today, they are freewares. they are open source tools. So you can download them freely and use them at wish. So you don't need any license to use them. Some of them may require you just to send an email and get a code to download, and that will just be it. So we can freely use them. At the end of this presentation, I will show us the links where you can download all the 9 software from. So the number 4 is ITK Snap. ITK Snap is another beautiful software that you can use for analyzing MRI images, most especially for brain images. That's what I do. I work a lot on brain images. And so this tool lets you do segmentation. It's particularly good in segmentation and in region of interest analysis as well. You can see this GUI showing to us the bottom left. You can see a subcortical structure there. And so you can see that it's very clean. This is T1 weighted images. And I know we have been learning a lot about T1 weighted images today. You can see that the image is very clean and the segmentation output is just spot on. You know, so ITK Snap lets you do segmentation and is very, very good at subcortical segmentation. And all the tools, they can work on 3D and some can do 4D. But in particular, ITK Snap does 3D and is very good at what it does. It's easy navigation for ITK Snap, and you just need to download it and play with it, and you will like it. So I've discussed about this. Yes, so the next one is MITK. MITK is another open source that you can use for processing MRI images. You can see that this lets you process more than brain images. So you can do anything that is MRI. It lets you import it. And I need to say that it allows you to import as analyze as well. And even I think as DICOM, you know, so you can do image registration, you can visualize your image, and it has a very good visualization interface. So that is MITK. Now, OSIRIS is the next one. OSIRIS, this is the user interface of OSIRIS. It's another tool that you can use for processing MRI images, different anatomy, and it lets you import DICOM images, and it can actually, it allows visualization of DICOM images. It's an image viewer. If you see, it says OSIRIS world fastest DICOM viewer. So this is more of a visualization tool than image processing tool. But when you have your DICOM images and you want to look at them before transferring them to where you process them, this allows you to do a very good job. There is a tool that I did not list that could be very, very useful for visualization too. It's called MRI Crow. MRI Crow. And there is a modified version called MRI Cron. You know, both of them, they are very good and they are very, very small. They are very small. So you can search for MRI Crow and you download it and it runs on Windows. So for easy visualization, you can use OSIRIS or you can use MRI Crow. Both of them are very, very good, portable and nice to use. Now I go to this beautiful software package called 3D Slicer. 3D Slicer, you know, allows you to batch process, even though it's GUI driven, but then you can automate whatever you do by selecting a folder and submitting what you want to process. And so it does segmentation very well, and it does registration. Time will fail us to go into details about, you know, what kind of registration, you know, is it, you know, a fine or whatever, but all the tools, they allow you to do registration for 3D images, and you can even do for 2Ds in some cases. So it allows you to do statistical analysis as well. That is 3D Slicer. It's very, very good. And then I will encourage us, if you want to do image analysis, you may need to look at 3D slicer. It's very, very easy to use. You just click buttons through, and then you are really home and dry. So batch processing, I've mentioned about it. So I'm trying to wrap up now because of time, and I know it has been a long session for us. So I want to talk about free software, and then we go to SPM. I did say that there are 3 most commonly used, and in my own opinion, the 3 most important software tools for processing and analyzing MRI images. So I did mention FSL, free software, and then an SPM. So this is free software. Free software was developed by a team in Harvard University. And so it's a very, very good software. The only thing is that it requires, a HPC or a machine with a very big, high capacity because it tasks the machine. So it's computationally intensive. And that is because the processes that it does sometimes, it targets to get, top or high accuracy. And that requires, a lot of, machine power, so that's free software. I've used it a lot, and it's particularly good when you want to do, maybe you want to extract the cortex, and it comes with its own visualization tool, which I have found very, very useful. Just like others, it's an open source. You can use it for segmentation. Now I'm aware that, in fact, a long time ago, free software and FSL, they collaborate, Oxford University and Harvard. And so free software and the FSL, they can work together. You can do part of your processing in free software and then go to FSL, or the converse. What I've had to do in the time past is because BERT, brain extraction tool of FSL, is particularly great. So I've had to do brain extraction in FSL, and I'll move to free software to do my cortical, extraction or subcortical segmentation. So, but it's just to say that it's possible for you to use 2-3 software packages to achieve a goal. You start with one, and then you can move to the next. And then the format at which you export your output in one can actually, let you, some of these tools, because they now work together, it will serve as input format in the other one. So free software is very good. As I mentioned, it allows you to do segmentation. It has a visualization module, and you can do registration as well. And just to mention that it can work in multimodal images, just like FSL and others. So you can use it for structural images, functional, resting state, diffusion, images, all these kind of images, largely, brain images. Free software can let you do anything you want to do. Now, the last but not the least is SPM, and it's a statistical parametric mapping, is called. So the original intention was to use SPM for voxel-based or voxel-wise analysis. So just imagine that you want to compare 2 groups. Maybe you want to see the effects of lithium in treating maybe those who have a psychiatric problem, and you have given them lithium for a period of time, and you want to see the effect in the brain. So sometimes you may be interested in voxel, level of analysis. So SPM will let you do that. So you group your images into those that you have given lithium and the control. And SPM is so clever to the extent that it will tell you the voxel where there are changes. So even if there is any activation in the area of fMRI, it lets you do it as well. So SPM can allow you to work on PET and on structural images. So it does a similar work to, although it's got its strength in the area of voxel-based analysis, but you can use SPM for registration as well, and it can work on time series images. So that's SPM, developed by the team in University of UCL, University College London. So SPM runs in MATLAB or on MATLAB. So you need a knowledge of MATLAB to use SPM. You can easily download it, and then I'll provide the link in a minute, and then you install it, and it's got a very nice user interface. It runs on Windows, it runs on Linux. Sometimes I have to get some of the functions and call them from Linux and use it for other things as well. So it works on all kind of MRI images. So in trying to wrap up, I just want to give the links where you can download these 9 tools. As I said, these are not the only tools that we use for processing or analyzing MRI images, but these are the most common ones in my view. And so FSL, as I said, is from Oxford University. So the link is there. The team is FMRIB, and then, but we typically call it FSL. And then number 2, the image, the link is there. You can see you can get it from NIH, website. And number 3, Amit from SourceForge. You can get Amit from SourceForge. And ITK Snap, the link is provided. So these are the links for the 4 of them. And as I said, they are freeware. So you can easily download them. MITK link provided. And for others, but just to call our attention to SPM and free software, you can see by the link that SPM is from University College London, and then free software is from Harvard University. In closing, I want to actually say that for all these tools, you can get materials online. If you want to learn how to use them, just self-paced study. There are lots of materials, like for FSL, for example. If you visit the website, you can download materials and read them. 1, 2, you can actually join the training, the universities or the teams that actually owns these software packages. They organize some training. And I'm sure in future, we will need to bring some of them to Africa to conduct training, because training are always ongoing by the developers of all these tools. So because there's many people use these tools and they're very, very useful. So I will say that visit the websites and see if there are training opportunities that you may want to actually take advantage of. So in closing, I want to say thank you to ISMRM as a big body, and to ISMRM Africa. It's really a great delight that ISMRM now has a footing in Africa. It has been a long dream, of many of us. And I want to appreciate the organizer of this meeting and those who facilitated, bringing ISMRM to Africa for the fantastic job they have done. I appreciate those who developed the software that I've shared with you today, because without them, no software. And if they did or developed all this software and they put price, then some of us will not be able to use. So they have made them available for free. So I sincerely appreciate them. And I appreciate you for listening. Thank you very much.